Hello, welcome to Physiological Psychology, the first part of our discussion on anatomy. This first section is going to give us a broad overview of how do we break down the nervous system and focusing mostly on the lobes of the brain and other major structures. When we talk about the breakdown of the nervous system, we're generally talking about the difference between the central and the peripheral nervous system. The central nervous system is the brain and spinal cord. When we talk about the brain, we usually stop there. The peripheral nervous system, however, contains a lot of function and structure, including things like our cranial nerves. We have nerves that are connected directly to the brain that are all through our head region, face, neck, even down to our gut. We have parts of the nervous system directly involved in digestion that function to a great extent on their own. Now, the main divisions of the peripheral nervous system are the somatic and autonomic. The somatic nervous system is things from our senses and muscles that travel to the nervous system, including our voluntary movement. Our autonomic nervous system are, the word autonomic sounds like the word automatic, and that's the best way to think about it. The automatic nervous system. The autonomic nervous system contains two major delineations, which are the sympathetic and parasympathetic. And the best way to think about the difference between those is parasympathetic, para like parachute. So the sympathetic nervous system speeds you up, involved in fight or flight, while the parasympathetic slows you down, involved in rest and digest. That's a traditional way of thinking about it, and it's certainly a good way to go. We look at the basic components of the brain, and the main things we talk about are the lobes. When we look at a brain, and coming from even an intro psych class, you're probably going to discuss the frontal, parietal, temporal, and occipital lobes. These are the four main lobes of the brain. They're visible when you look at the side of the brain. And the delineations between them are kind of visible, like more visible than other things. Now, the temporal lobe is separated from the rest by that deep sylvian fissure between the temporal lobe and the frontal lobe and even part of the parietal lobe. Right? The frontal lobe here in blue, above and in the front, right behind your eyes. Now, the frontal lobe is separated from the parietal lobe by the central sulcus. This is a long indentation that runs down the length of the brain between the frontal and parietal lobes, stopping above the temporal lobe. And it is fairly visible on human brains. Not all of them are the same, and that's an important characteristic too. The brains we're looking at are what we would call like a canonical brain, kind of like this is how we expect a brain to look. But when we look at a real human brain with all the rises and valleys, the gyri, the raised areas, and the sulci, the indentations, there's a lot of variability between brains. The occipital lobe in the back of the brain is separated. There's the parieto-occipital sulcus that's only visible from the inside, and on the outside it's not entirely clear where the occipital lobe begins or ends and the parietal lobe begins. We also have our cerebellum, which is visible. At the bottom of the brain, you see the darker area. The cerebellum is like a little brain. It looks kind of like a miniature version of the brain. As we've discussed previously, it contains the majority of the brain's neurons. And has kind of like a smaller version of it. And then it's right above the brain stem. The small delineations help us to find our way around to some extent, and also help us to understand when we're talking about the brain, we're going to see these terms quite often. Lamina. Well, lamina are layers. And so lamina, these laminae and these columns organize brain function. Very visible in the occipital lobe, but throughout the brain, they're there. A tract is a pathway. It's generally a lot of nerves together And so major connections are often called tracts. Nerves are the nerve fibers themselves, right? Bundles of those. Nucleus is an interesting term. When we think of nucleus, we think of the nucleus of an atom, or we think of the nucleus of a neuron. When it comes to a nucleus in the brain, we're talking about a center of function. So the brain has a number of what we call nuclei, right? An area where there's a lot of function, especially in densely packed areas like the hypothalamus. You'll see a lot of these. A ganglion is a nerve connection, and then a gyrus or sulcus are our raised and lowered areas in the brain. The wrinkles of the brain. The top part's the gyrus, the bottom part's the sulcus. Now let's start with the laminae and columns, because these are concepts that are often overlooked. But when you look at the brain under a microscope, when you look at the surface, right, so you've got your sulci and gyri, these raised areas and these indentations, it's all surface of the brain. Whether you're at the top of the hill or the bottom of the valley, you're still at the surface of the brain. And your sulci and gyri, the wrinkliness of it, creates more surface area. If your brain was completely smooth, and there are rare neurological disorders like this, if you were born with a smooth brain and it never developed wrinkles, you would not be functioning. You would be severely, severely developmentally delayed and never really reach a typical area of function, because these the higher level functioning, a lot of the brain is in these sulci and gyri. The surface is the gray matter, and then way down deep is the white matter connections, the nerve connections, right, your tracts. Now, your surface of the brain contains these columns and laminae. So, originally really discussed in regards to the occipital lobe, in the occipital lobe, you can clearly see 6 layers of neurons. And when you look under a microscope, you can see that they're organized in this way. These images here are more kind of like generalities. The idea is, in the 4th layer is your inputs from elsewhere. So you've got neurons, axons ending here, coming from all over the brain for processing. Your levels 1, 2, and 3 are higher levels of processing, and then 5 and 6 are outputs in connections with other areas. They're also arranged in columns. So if you think of a line in a particular orientation, the size, the length, the characteristics of this line are all going to be processed in a particular column in the occipital lobe. The columns are going to be similar. So you might have columns that have lines with similar orientations that are near each other. So when you view something, this is all getting pulled apart into the components that are being detected by our visual sensory detectors, and then they go to the back of the brain to be put back together. And then as we move out, we get more and more complex in terms of processing. But the idea is your brain is organized in these columns and laminae. And so these columns have similar function, and these, all the cells within a column do similar things. And then laminae are these layers that also do, very specific tasks within those columns. When we look at the brain, we're looking at what we would call these different views. So traditionally, when we look at a brain, we're either looking at the surface or we're cutting it in half and looking at the inside. Like the most detailed views are going to be cutting the brain in half, because in the middle of the brain is where you have these deep, important structures that are involved in our neurological functioning. So this is called a sagittal plane or a sagittal cut. The frontal plane is also called the coronal view. And coronal, it means crown. And the best way to visualize this that I've ever seen is you take a crown, you put the crown on your head, and then you go straight down. Right? That's coronal. Right? Coronal, crown, cutting in half between front and back. The transverse plane is also called the horizontal plane, and that is taking slices this way. So when someone has a brain scan done, often they will take these transverse slices from the bottom to the top and look at, for instance, things like a CT or an MRI in this way. The most common ones we're going to use, besides looking at just at the surface of the brain itself, is going to be the sagittal plane, because it gives us a lot of information, and it's the kind of traditional way of looking at it. But it still doesn't show us everything. Now, if we take the surface and we take a sagittal view, that gives us most of the structures that we care about, but not all. And that's the idea. It's not going to give us everything. We're doing the best we can to view as much of the brain we can without getting bogged down. Now, some more anatomical terms. Traditionally, we would talk about Brodmann areas. Brodmann was a researcher in the late 1800s, early 1900s, who looked at the brain under the microscope and kind of divided it up based on the way cells were configured. And there were tissue differences that he found, that he said this is clearly, these areas would be next to each other, but they're clearly different. And the idea was they're probably performing some kind of similar task that's different from something else, which is really kind of an interesting thing. Well, the problem is these numbers are all over the brain. It's a little hard to, you basically kind of have to memorize where they are, what they are, but it doesn't really tell us anything in terms of functional anatomy. And in PhysioPsych, the number one thing we care about is functional anatomy. Where are you and what do you do? And so we use terms to help us find our way around. If I say dorsal, I'm talking about the top of the brain. If I say ventral, I'm talking about the bottom of the brain. Dorsal means back and ventral means belly. And our brains, our heads, are on different than pretty much every other mammal. The idea is, think about a dolphin, right, with a dorsal fin on its back. Its head is still pointing this way. That would be like us. Here's our our belly, our back. That would be like our head pointing this way to be in that same alignment. So our plane actually shifts as it comes up our spinal cord, and then kind of our brain resting on top of it in a different kind of way. So dorsal is the top of our brain, the back. Ventral is the bottom of our brain, the belly. And so we can talk about the dorsal view, the ventral view, the dorsal stream, the ventral stream. We also talk about, we use terms like anterior and posterior to help us find our way around. Once we know our four main lobes, we've got our, say, all right, we want to talk about Broca's area in the frontal lobe. Well, it's going to be the inferior posterior part of the frontal lobe that helps us find a way around. So you say, oh, here's a frontal lobe, we're looking towards the back of it, and we're looking towards the bottom. And that's the idea. Anterior is toward the front, posterior is toward the back, superior is above, inferior is below. And this image is interesting that I'm looking at it because this is the right side of the brain. And for most people, our Broca's and Wernicke's areas are actually on the left. Now, we got those. Medial means toward the center. Lateral means toward the side. Ipsilateral means the same side as, and contralateral means the opposite side. Well, this term, contralateral, is the idea that my right hand is controlled by the left half of my brain. Right? The left side of my brain contains the motor programs for the right side and all the connections to the right side of my body. Contralateral, opposite side, versus ipsilateral, same side. Our spinal cord requires a little bit of consideration here because, remember, in terms of reflexes, your spinal cord is basically doing brain stuff. It's doing processing. Now, in our brain and our head, we've got the gray matter on the outside, the higher levels of processing, and the white matter tracts inside, making connections. In our spinal cord, we actually have the gray matter on the inside, surrounded by the white matter tracts. And so you also have these pathways, right? And so information works like this in the spinal cord. Dorsal is input sensory, ventral is output motor. So dorsal is coming in towards the back, and ventral is out the front, right? So our kind of sensory stuff is going in toward our back part of our spinal cord, goes up to our brain for processing, comes back down, and our motor stuff is coming out the front. So we've got all these nerves that are connected, And that's the way they're organized. Now, here's another example of the delineations of our nervous system. And what we're looking at here is our main ways of splitting things up, right? Our brain, and this is from our text, our nerves, our digestive tract, and we see here ENS. Well, we've got our peripheral nervous system, which contains our autonomic nervous system, which contains our sympathetic and parasympathetic nervous systems. While our digestive tract, the enteric nervous system, is a fairly new term. It's been really more researched recently, and it's shown, for instance, a couple interesting things. One, the majority of the serotonin in your brain and body is actually in your gut. And two, it functions pretty much on its own. And there's some really interesting connections between the gut and the brain. And there's a lot of research now about health and about diet, about how we treat our gut has a direct and important effect on our brain and our overall health. So it's a very interesting new area to look into. Now, the autonomic nervous system, we said this is our gastrointestinal part. When we look at the traditional sympathetic and parasympathetic, we're talking about heart rate, pupil size, digestion, respiration, and sexual response, things that we don't necessarily consider conscious control over, right? When you're afraid, your heart starts pounding. You don't think, oh, I'm afraid, I need to get ready to act, right? This is an automatic process. And the primary neurotransmitter involved in the sympathetic nervous system is norepinephrine. And for the parasympathetic, it's acetylcholine. And we recall from our neurotransmitters how these things function. Now let's go and look at the brain itself. We're looking at, on the left, a fish brain, right? And in the middle, we've got an embryo brain. And what we're kind of considering here is these major delineations. All right, so in a fish brain, there is no real cortex, very, very little cortex there. Their telencephalon is tiny. They do vision, they do the movement stuff, decent sense of smell, very little higher orders of processing, right? And if you've ever seen a fish brain, it's tiny for most of them. And then for your embryo, what you should notice is the ratios of these things are really quite similar. A fairly small telencephalon and diencephalon, which contains your kind of thalamic nuclei and areas there. And then you get your hindbrain, right? Your rhombencephalon, which contains the pons, medulla, cerebellum. So you're looking at this and you're saying, okay, you've basically got in a 6-week embryo brain, not terribly different in terms of, spacing it out as these kind of like very simple kinds of creatures. And you also have a tail at this point too. So you're looking at this really interesting kind of evolutionary holdover in terms of how the brain is organized. Now, one of the things we're also going to see is in some of these simpler organisms in early human development, we really have a larger portion of the brain being this midbrain, right, this mesencephalon. And yet, as we become adults, these things take up less and less overall real estate in our brains, right, where we are lots and lots of telencephalon and this kind of that cortex. What we think of when we think of like what makes a human brain special, the cerebral cortex, the newer stuff, and the higher levels of processing. Our olfactory bulbs, fairly small, compared to something like the proportion for like fish and other creatures. This part of the autonomic nervous system is responsible for slowing heart rate during rest. Is it central nervous system, somatic nervous system, sympathetic nervous system, or parasympathetic nervous system? And if you recall, the autonomic nervous system has two delineations, sympathetic and parasympathetic, and this would be the parasympathetic. This part is responsible for fight or flight. Well, since we already said the autonomic nervous system is sympathetic and parasympathetic, and we said that parasympathetic is involved in rest and digestion, or specifically rest, then this would leave us with the sympathetic nervous system. Now let's take a look at how we are with our terminology. The parietal lobe is what compared to the temporal lobe? Like what to the temporal lobe? Is it lateral, anterior, superior, or inferior? Well, let's consider what this means. Parietal lobe is kind of above it, right? So you've got your brain, you've got parietal lobe above your temporal lobe. So you've got lateral means to the side, it's not to the side of. Anterior means it's in front of, not really, no. Superior means it's above, and inferior means it's below. Well, since we said the parietal lobe's here, temporal lobe's here, we're going to go with the parietal lobe is superior to the temporal lobe. And we could say, conversely, the temporal lobe is inferior to the parietal lobe. Now, here are some more terms in the central nervous system. The cerebral cortex being this nice kind of wrinkly part on the outside of the brain, the big part of the newer part of the brain. The longitudinal fissure. We can't see it from this image, but if you take a brain, right, and you look on maybe from the top, you can see, like, if this was the brain, okay, the longitudinal fissure would be right separating the two hemispheres. The two hemispheres are connected, right, left and right hemisphere, are connected by the corpus callosum, a large band of white matter that connects the left and right hemispheres and basically causes them to function as a single thing. Without a corpus callosum, if you were to have your corpus callosum severed, you really would have two conscious selves in your head. The limbic system we also can't see from this image because it requires a sagittal view. Includes things like the thalamus, hypothalamus, and related structures. Your central sulcus is separating your frontal from your parietal lobes, and the lateral sulcus is this one that is also called the sylvian fissure, separating your temporal from your frontal lobes. So these are great landmarks that allow us to say, look, if I know where the central sulcus is, and I say precentral gyrus, that means I am the gyrus right in front of the central sulcus. I'm the very posterior part of the frontal lobe, and I do very specific tasks involved in movement. Now let's move through the brain, first starting with the hindbrain, and then moving forward into the lobes. So we start with these three things, the medulla, the pons, and the cerebellum. Well, the medulla is involved in a number of tasks, but it's kind of like this area where, you know how like the left side of my body is controlled by the right side of my brain? This is also where these tracks cross. This is in the posterior portion of the brain, above the spinal cord, and the brainstem consists of the hindbrain structures, the midbrain, and other central structures of the brain. The midbrain being basically right above these structures and involved in some automatic processes. Think of it like this, is the deeper you go in the brain, the more automatic these processes are. You know, when you hear a sound or see something move and you go to look and see what it is and it happens automatically, this is basically what has sometimes been called like the lizard brain, which is an okay description, where things just kind of draw your attention and it's not your level of conscious awareness that makes it happen. Well, your medulla, we mentioned, is a large extension of the spinal cord where these tracks cross over. It's also responsible for things like breathing, heart rate, vomiting, salivation, coughing, and sneezing. These automatic processes involved in survival. Why do we vomit? We vomit to keep ourselves from dying. If you eat something that's bad, your body is going to have an automatic response to protect you. You also have your cranial nerves. Well, our cranial nerves is something we'll get into. But you have these controls for the head, your muscle movements, and a number of outputs. So you've got cranial nerves that are connected directly to these deep parts of the brain. In fact, the majority of the cranial nerves, the majority of these peripheral nervous system connections that connect directly to the brain connect here at the medulla, the pons, somewhere deep. Not all of them, but many. Now, starting with the back of the brain, we start with the occipital lobe. It's located at the posterior portion of the cortex. And traditionally, it's been called also the striate cortex because it looks fairly striped under magnification. Remember those laminae, those layers? Well, that's what makes it look striped. In a close view, these layers are very distinct, specifically in the occipital lobe, less so in other parts of the brain. Your occipital lobe basically does vision. Where other parts of the brain have multiple tasks, vision is such a massive component of what we do that when it comes to processing here, it's dedicated. In fact, it will even shunt some of these vision processing tasks up to your parietal lobe and down into your temporal lobe for further processing. So it's heavily involved in this, basically what it does. The tracks come directly from your receptors in your eyes to your thalamus to your occipital lobe, with those tracks being your optic nerve. Cranial nerve number 2 connects directly to your eyes, travels to your thalamus for processing, and then is shipped to your occipital lobe for further analysis. If you have damage to your occipital lobe, you could have perfectly functioning eyes and connections to your thalamus. But if your occipital lobe is damaged, you can have what is called cortical blindness. Sometimes people are blind due to cataracts, due to eye damage. In this case, it's the brain that is damaged that is causing this cortical blindness. As we go up, right, from the temporal lobe to towards the forward part of our brain, we enter the parietal lobe. This contains the post-central gyrus, which is our sensory information. The idea being, when you detect sensory input, this is where it's processed for body, right? Where is your body in space? What is your body feeling? Where have you been touched? We also get processing from muscle stretch and joint receptors as part of proprioception, which is a sense involving where is your body in space? We're going to integrate information about where our body is with our visual and all these other components. You think of an athlete, like a stunt athlete, or who has to kind of know where they are and integrate a lot of information. Think of someone like a race car driver or a fighter pilot who is moving so quickly and they have to input all of these things. It's all going where their body is in relation to their senses. We also process spatial and number information primarily in our parietal lobe. So we think of sensory, body, space. This is a good way to think about our parietal lobe. Now, as we move forward, we are now looking at what we call the homunculus. And this is an old term. It basically means little man. And there's some old models from people like Wilder Penfield who they actually say, argue that you can really see these enlarged areas in terms of function, right? So what do we have? What are we very sensitive to? Well, if we look at the sensory component here, it's on the left, we see that our hands are very sensitive and our faces are very sensitive. A large portion of sensory processing is here. The trunk of our body, very little. It doesn't do a lot of information for us, right? And that's parietal. And then frontal, right next to it, just on the other side of the central sulcus, where your somatosensory strip is your post-central, behind the central sulcus, and your motor area is your pre-central, in front of the central sulcus. And we look at motor. Very similar in regards to how much processing we do. Our hands, both sensitive and a lot of control. Our mouths, our faces, our jaws, our pharynx, larynx. Well, you think about how important it is for both sensory and motor for this. Speech, breathing, chewing, drinking, we're using the same piping for all those tasks. And certainly, you've had a conversation at a meal before, right? You're out with friends, you're eating together, you're talking, you're laughing. You're coordinating, your body is working hard to coordinate this so you don't choke on your food, right? Or drown yourself when you drink. Very carefully coordinated. Now, before we move, and it should make sense that sensory and motor are right next to each other for ease of communication and ease of processing. We want these things to happen fast. So a lot of knowing where things are in the brain helps us to understand how the brain works. If you know what's next to what, you know what it does. I've heard it said about the world that geography is destiny. And that may have a great effect, but it's definitely true in the brain. When it comes to the brain, what you're next to is what you do, right? It's related very heavily to what you do. Well, the temporal lobe, before we go into the frontal lobe, here is our primary auditory cortex, right? At the superior portion of the temporal lobe is our auditory processing. And our temporal lobe also folds kind of under, and if you actually have a brain, you can kind of pull it back a little and look in there. There's even more cortex in there. There's more surface of the brain that's not on the surface of the brain. I know that sounds weird, but brain surface is so important. We've actually got these like nooks and crannies in the brain. It's called the insula. That's inside that sylvian fissure where there's more cortex. Well, we have our left and right auditory cortex, right, processing information, and it's also responsible for complex parts of vision, including movement and some emotion and motivational behaviors. So there's a lot more happening here. Kluver-Bucy syndrome happens when people have damage because inside your temporal lobe, you have your hippocampus and your amygdala. So kind of like embedded in there with temporal processing around there. Well, your hippocampus does memory. The hippocampus is your primary driver for putting your experiences into permanent storage in your brain. Your amygdala helps your hippocampus to know what's important. Things have an emotional connection for you. For instance, you may not remember going to the store every time you've gone in the past year. Because why would you? It's not important. But on any of those trips, did you meet somebody? Did you maybe drop something or forget something that was a problem for you? Let's say you met somebody and you had a really nice conversation with somebody you found attractive. You're probably going to remember that trip because all of a sudden you've got your amygdala saying, this is important, right? And so it goes into your memory. But things that aren't important, your memory is just not going to keep them. So that's part of this. Now, Kluver-Bucy syndrome is for people who have damage to their amygdala. And all of a sudden, they have issues with feeding, right? They start eating non-food things. Their sexual behaviors change. And they become placid to a certain extent. They don't have an emotional involvement. So we can see that with temporal lobe damage. And the ventral pathway from the occipital lobe in terms of vision, what is it, right? So the dorsal pathway is for the parietal lobe. Information goes up, and you say, where is something in space? For the ventral pathway, information goes down to the temporal lobe, and you say, what is this thing? I show you a picture of a hammer, right? And I say, what is this? I don't tell you what it is. Up, where is it in space? What does it look like? What does it, appear to be? Down, what's it called? Right? What is this thing? Well, temporal lobe, heavily involved in language and naming and things like that. And it makes sense because your auditory cortex is there, and you're very close to Wernicke's area is part of you, and you're still pretty close to Broca's area too. Broca's area being in the frontal lobe. So here we have a couple of key terms. I mentioned the precentral gyrus. Here we do motor. So the frontal lobe, we often talk about things like personality, motivation, but we also understand things like motor, which again is going to be related to that, right? Motivation and action and doing things. Well, the precentral gyrus is our primary motor cortex, and we have a lot of associated motor areas, including, eyes, our eye function. We also have our prefrontal cortex, which is broken down into more components. And each of these lobes will go into far more detail as we go on. But the key to anatomy is knowing basically what are these things and what do they do. We have to know what these things do. Right? Now, before we go into the prefrontal cortex, let's look at, can you tell me about the lobes? This lobe is associated with auditory information. Is it frontal, parietal, temporal, or occipital? And it's the temporal lobe. This lobe is associated with sensory information. Where does our sensory information go? Which lobe processes this? Frontal, parietal, temporal, or occipital? And importantly, this lobe, in addition to processing sensory information, also tells us about space and navigation. And that would be our parietal lobe, also part of that dorsal pathway information, visual information from the occipital lobe going up. The central sulcus separates the frontal lobe from the what? Okay, now this puts it together. We've got a frontal lobe. What's it next to? And what's it getting separated from by the central sulcus? Well, the frontal lobe is next to the parietal lobe, and it's next to the temporal lobe. Right? It's above the temporal lobe and next to the parietal lobe, in front of the parietal lobe. Well, the central sulcus is running from the top down, separating the frontal lobe from the parietal lobe. And the sylvian fissure is separating the frontal lobe from the temporal lobe. So in this case, the frontal lobe is separated from the parietal lobe by the central sulcus. Now, the prefrontal cortex is one of the cooler things that we talk about in terms of what the brain does. Now, if you remember from intro discussion of lobotomy and the monster Walter Friedman. Walter Friedman being this hack physician who started to popularize lobotomy, what was called the ice pick lobotomy, by having patients come in and making them unconscious, usually through electroshock or something, and then lifting up their eyelid and jamming an ice pick behind their eye, tapping it through with a hammer, and then wiggling it around to destroy as much of the prefrontal cortex as possible. And when they were done, these people had severely reduced IQs, and they were very easy to deal with. So this was often done for people who were just difficult to deal with. Maybe they had psychiatric disorders, and for Friedman, doing them all over to thousands of people, right? Most of whom would have done much better from other treatments. It was really world-shattering because it changed people permanently. And it changed people because in our prefrontal cortex, right, the primary target for these things, these higher abstract functions and these things like working memory. And people with damage here show delayed response. They're slower. We also see with frontal lobe damage, frontal lobe syndrome, a lack of motivation. People, they just don't do anything, right? It's hard for them to initiate. And so that's a big function here. So the prefrontal cortex, being part of the frontal lobe, is one of the more interesting areas in regards to kind of who we are in our sense of self. Well, that's the first part of our anatomy discussion, and we're going to go into more depth into more of the brain soon.